Hello, my dear students. Hope you are doing great and have a good time. I am Bal Rishi, your biology educator. As we know that we have started molecular biology of inheritance. In molecular biology of inheritance, one of the most important topic as per NEET point of view is that gene expression. Firstly, we will explain what is meant by gene expression. I will firstly here write the meaning of gene expression. The definition of gene expression is how gene expresses. How gene expresses. This is a simple definition of the gene expression. In other words, we can also say that means gene forms proteins to express themselves. That means we can say that the genes do not directly express themselves, but how to express themselves, the process is that genes form proteins to express themselves. This is the definition of the gene expression. That means, in simple language, I will explain it by a simple example. Let us, my dear students, you have qualified NEET. This candidate qualified NEET. This kid qualified NEET. When it qualifies NEET, the expression of happiness can be expressed, can be vivid, observed from his face. That means his facial or her facial expressions determine the happiness. But the person is internally happy, as well as the expressions are shown by his or her face. Simple is the procedure here, simple is the process also here. That means gene is directly do not express themselves, or gene does not express itself, but it expresses by synthesizing proteins, and when it synthesizes proteins, these proteins are meant for expression. Dear students, this is a simple definition of the gene expression. But how to control, as per NCERT, how to control the gene expression? Firstly, why we need control? There are some questions why we need control, what is meant by control, and what are the different levels of control in our body or in a living organism, that we are going to discuss here. And firstly, what is meant by control? The definition of the control is that, as we have already learned about, genes form proteins to express themselves. That means genes result is the formation of proteins. That means genes synthesize proteins. What is meant by control? That means simple genes or a simple gene forms protein. It synthesizes protein. We have to control the synthesis of protein. This is known as control. That means simple definition is that means control of protein synthesis. What is meant by control? That is protein synthesis control. How to control? This is known as control. That means how to control a protein synthesis. How we are going to control a protein synthesis? This is known as control. But we know that there are the two living organisms as per NCERT. That means we have two different types of the cell in two different living organisms. That is prokaryotic cell and eukaryotic cell. But we have to determine the levels of control in prokaryotic cell as well as in case of eukaryotic cell. Are you getting me, dear students? That means this control, we have to control the synthesis of proteins. but we have to control the protein synthesis in case of prokaryotes, and we have to control the synthesis of proteins in eukaryotes or eukaryotic cell. Now, dear students, this is levels of control or level of control. Level of control. There are the two levels of control, that in case of prokaryotes and in case of eukaryotes. Firstly, we will take the case of prokaryotes. What happens in prokaryotes? What happens in prokaryotes? How proteins are synthesized in prokaryotes? We have to. There is a particular pathway. We have to discuss this pathway. There is a particular pathway by which proteins are synthesized. That means genes are present on DNA. That means this DNA results, or from this DNA we are synthesizing RNA, and from RNA we are synthesizing proteins. This is the pathway for the synthesis of proteins in case of prokaryotes. This is the pathway. Dear students, this DNA, from when the DNA synthesizes RNA, this process is known as transcription. This process is known as transcription, and the process by which proteins are synthesized by the from RNA, this is known as translation. This is known as translation. That means, dear students, here the two processes occur for the synthesis of proteins. For the synthesis of proteins, we require transcription and translation in case of prokaryotes. Clear? Now, this means there are the two setups. Setup number 1 and setup number 2. Now, we are going to discuss the levels of control. We are going to discuss the control where we are going to control the synthesis of proteins. Protein synthesis control. That means control the synthesis of proteins. There are the two setups in case of prokaryotes, that is transcription and translation, where we can control the synthesis of proteins. Isn't it? Are you getting this? Yes. That means this synthesis of RNA from DNA is known as transcription, and synthesis of proteins from RNA is known as translation. Now, there are the 2 points where we can control the synthesis of proteins. Now, dear students, for the explanation of the process, for easy to determine how there occurs a control, we here draw a diagram of prokaryotic cell. This is the diagram of prokaryotic cell, and this is the diagram of prokaryotic cell. In prokaryotic cell, we know that the nucleus is membrane-less. That means nuclear matter, genetic matter, DNA is directly put into the cytoplasm. It is directly put into the cytoplasm, and from this DNA, we are going to synthesize RNA. And from this RNA, we are going to synthesize proteins. This occurs in case of prokaryotic cell. And the synthesis of RNA is setup number 1, and the synthesis of proteins is setup number 2. Dear students, we know that in case of prokaryotes, the synthesis of RNA, the transcription and translation both occur in cytoplasm. Thus, what to control, whether at a point 1 or point 2 or at both points? No, it is the control at a single point. Why there is only a control at a single point? That means we can control at setup number first only. Why we can control the synthesis of proteins at setup number 1 only? Dear students, as we know that prokaryotes, transcription and translation is a coupled mechanism. That means when transcription occurs, at the same time translation occurs. Thus, that is the reason why we can set up or we can control the synthesis of proteins at setup number 1 at transcriptional level, not at translational level. That means when transcription occurs, that means 100% sure we are here synthesizing proteins. That means we can't sit up at setup number 2 because this is a coupler mechanism. Because these two processes occurs continuously, simultaneously. That means that why we control at setup number 2. The reason is that what is the reason? What is reason? Because both process, setup number first and setup number second, transcription and translation occurs simultaneously. These occur simultaneously. When transcription occurs, that means translation is 100%. Translation is sure. This is the setup where we can control, that is transcription in case of prokaryotes. That means in simple way, we can here control at setup number 1 only at transcriptional level. This is the control in case of prokaryotes. Now, what happens in case of eukaryotes? What happens in case of eukaryotes? This is about eukaryotes. What happens in case of eukaryotes? Simple process is also here. That is, we have to make a control at a particular point from which we can either control the synthesis of, or increase the synthesis of protein, or decrease the synthesis of protein. That means simply we are going to control the synthesis of proteins. That means in case of eukaryotes, firstly here we are going to write this pathway by which proteins are synthesized in case of eukaryotes. The procedure here is that DNA, from DNA we are going to synthesize HNRNA, heterogeneous nuclear RNA. From this we are going to form RNA, and from RNA we are going to synthesize this protein. This is the pathway. Now we will describe this pathway with the help of a diagram. This is the diagram by which we can experience the synthesis of proteins in case of eukaryotes, or we can here describe the control of protein in case of eukaryotes. Dear students, let us consider this is nucleus. And in case of eukaryotes, the DNA is present inside the nucleus. This is DNA. Dear students, this is DNA which is present inside the nucleus. Now what happens? From this DNA, we are synthesizing heterogeneous RNA. And from this heterogeneous RNA, we are going to synthesize RNA. This is the process. That means the first number, setup number one is simple transcription. Setup number 1 is transcription. We are here synthesizing HNRNA from DNA. This is setup number first. Setup number 2, when we are synthesizing HNRNA, when we are synthesizing RNA from HNRNA, this is known as, this setup number 2 is known as post transcriptional modification. This is the post transcriptional modification. That means setup number first, setup number second. These are the first two setups. The second setup is also known as processing, or we can here write processing. This is also known as processing. Now what about RNA? What is the third setup? The third setup is that we have to transport this RNA from nucleus to outside in cytoplasm. We have to transport it outside because the synthesis of RNA or HNRNA occurs inside the nucleus, but from the synthesis of proteins from RNA occurs in case of cytoplasm. Thus, we have to transport it from nucleus to outside chamber, that is outside inside the cytoplasm. And the third step is transport. The third step here is transport of RNA. Transport of RNA from nucleus to cytoplasm. From nucleus to cytoplasm, we have to transport RNA. And final step, this is the third step, and final step here is that means translation. Translation. What is meant by translation? Translation is a process by which we are synthesizing proteins from RNA. Now, in case of eukaryotes, there are four steps. Number one step is transcription. Number two step is post-transcriptional modification. Number third step is transport of RNA. Number fifth step is translation. Where to control? Now, where to control? In case of prokaryotes, we have step number one is the control, where we can control the synthesis of proteins. But in case of eukaryotes, there are four steps where we can control the synthesis of protein. Is it? Are you getting me, dear students? Are you getting me, dear students? That means these are the four steps. We can control at step number one. We can control it at step number two. We can control this at step number third and step number four. These are the four steps where we can control this cross synthesis of proteins in case of eukaryotes or in case of eukaryotes. Are you getting me? 1 second, dear students. 1 second. Now, dear students, we have learned about the levels of control in case of prokaryotic cell and in case of eukaryotic cell. Now, the second question is why we need control. The second question is that why we need control. Why we need control? For what purpose we are going to control the process by which proteins are synthesized? Why we need control? Why we need control? Question is why we need. Why we need control? Why we need control? There are few reasons by which we can explain why we need control. There are few reasons by which we can explain why we need control. Dear students, in case of prokaryotes, we have learned there is a single point where we can control, that is translation, transcription. But in case of eukaryotes, there are the 4 points where we can control the synthesis of proteins. Then why we need control? Number one reason is that is metabolic need. That is metabolic need. Metabolic need. Metabolic need. What is meant by metabolic need? That means metabolic need when there is more BMR. When we require more BMR, at that time we have to synthesize more enzymes, we have to synthesize more proteins by which we can increase BMR. We have to synthesize more proteins these are, for example, enzymes which can digest more food. We can increase our BMR rate. And second point here is that this is the first point. Second point here is that physiological need. Physiological need. This is the second point. What is meant by physiological need? Simple case, that means there is different physiology and different adaptations. That means when a human being which is present, which lives in tropical areas, which lives in hot desert, which lives in the polar areas, so on and so forth, these physiological needs are different among the different organisms, or different adaptations need different physiological needs. And number third, dear students, is that is developmental need. Developmental need. Developmental need. What is meant by developmental need? Dear students, we know that up to the puberty our body is normal. After that there occurs simple slight change. there occurs a slight change in our body. There occurs the development of breast, there occurs the development of muscles, there occurs the more development of bones, there occurs the development of beard. That is secondary growth. For very much secondary growth, we require more BMR, we require more hormones. For the production of hormones, we require more enzymes. For the production of these enzymes, we require more, that means expression of genes. That means this is the reason why we need control. Up to the level of the puberty, we don't require such enzymes which are synthesized at the time or after the puberty. Clear? Are you getting me? That these are the three reasons why we need control. Now, dear students, we have different types of the genes. We are making different types of genes here. That means first type here is that types of genes. On the base of control. Types of genes on the base of control. Types of genes on the basis of control. Dear students, we have different types of genes on the basis of control, on the basis of function, so on and so forth. Firstly, we will describe here types of genes on the basis of control. There are the two types of genes. On the basis of control. There are the two types of the genes on the basis of control. Number one gene is constitutive gene, and number two gene is non-constitutive gene. Constitutive gene and non-constitutive gene. These are the two different types of the genes. Constitutive gene, that means we can here write CG, and for non-constitutive gene we can here write NCG, short form. Constitutive gene and non-constitutive gene. Dear students, on the basis of control, these are the two types of the genes. That means what is meant by constitutive gene and what is meant by non-constitutive gene. Constitutive genes are those genes which are never under control. That means they do not require any control. And these genes are under control. Non-constitutive genes are those genes which are under control. That means constitutive genes do not require any control. Non-constitutive genes require control. That means simple process is that in simple layman's language we can say that these are those genes which are continuously working, which are continuously synthesizing proteins, which are continuously synthesizing enzymes, which are continuously synthesizing their products. Clear? That means these constitutive genes are working continuously throughout the life of an organism. Clear? These genes are also known as housekeeping genes. Let me explain it with you by simple example. We know that health sector is one of the most important sector, and in case of health sector, the medical faculty, the medical doctors and medical practitioners must be present 24/7. Must be present in hospital or at their particular point of hospital or clinic. They must be present 24/7. Similar case is here. These genes are working 24/7. These are working 24/7, and these are working at a particular point. These are working at a particular time. That means, for example, let us take the example of follicle stimulating hormone. FSH, follicle stimulating hormone. This follicle stimulating hormone is released at the time of puberty. From puberty onwards, this follicle stimulating hormone is released. And before the onset of puberty, this FSH is not released. And when the FSH is released at a particular time, this gene means this type of gene which produces, that means FSH is considered under non-constitutive gene. And for glycolysis, for the formation of tRNA, and do not require any control for the formation of, for example, in case of glycolysis, We do not require any control, and this type of genes, these type of genes are known as constitutive genes or housekeeping genes. Clear? This is the one type of the gene, and next type of gene is on the basis of function. On the basis of function, genes on the basis of function. On the basis of function, there are two different types of the genes. On the basis of function, there are two different types of genes. Number one gene is known as regulator gene. Number one gene is known as regulator gene, and number two gene is known as structural gene. Number two gene is known as structural gene. These are the two types of the genes, that is regulatory gene and structural gene. Let me explain this regulatory gene and structural gene with the help of diagram. Dear students, I am here going to draw a diagram by which we can simply explain this regulatory gene and structural gene. Simply we can explain here. Let us consider this gene is regulatory gene and this gene is our structural gene. For the regulatory gene, we require a site, that is promoter site, we require terminator site. For the structural gene, we require promoter site, we require terminator site. Let us consider in case of E. coli, we know that there are ZYA. That means, for example, like operon, we are discussing that like operon model. Here this is the regulatory gene and this is the structural gene. And this structural gene forms enzymes. This form enzyme and this form enzyme. Let us consider this enzyme 1, enzyme 2, and enzyme 3. That means these three systems, these structural genes result in the formation of enzymes. Are you getting me? Similar is the case with here. That means from this regulatory gene, we are here firstly forming messenger RNA. Also in case we are forming different types of RNAs, messenger RNAs from E1, from C1, C2 and C3. We are here forming messenger RNA number 1, messenger RNA number 2, messenger RNA number 3. Simply here we are also forming messenger RNA, and from this messenger RNA we are here forming protein. Now what happens? Let us consider this is the shape of protein. If this protein comes and binds here and stops the action of structural gene, then this gene is known as regulatory gene. And when the regulatory gene stops or controls the structural gene, this is known as regulatory gene, and on which it controls, it is known as structural gene. That means what is meant by regulatory gene? Controls other genes. Regulatory gene is that gene which controls other genes. And what is structural gene? Which is under regulatory gene, under the control of regulatory gene. Which is under control of regulator gene. This is the structural gene and this is the regulator gene. Clear? Now dear students, we are here to describe two types of genes on the basis of their function, regulatory gene and structural gene. Now, dear students, what about the regulatory gene? The regulatory gene is here forming a protein. And this protein either inhibits or enhances the activity of structural gene. Depending on that, we have two more types of genes. That means this regulator gene, this protein enhances or it decreases the activity, then it is negative impact or it has it is positive impact. On the basis of that very much impact, we have number one negative. We have number one negative control. Actually, these genes control, this regulatory gene controls the structural gene. Now this regulatory gene controls, that which decreases or it is negative effect on the structural gene or it is positive effect. Then on the basis of that we have negative control, and number two, that is positive control. Negative control and positive control. What happens in negative control? Let me explain. This is regulatory gene. Dear students, kindly have a look here. This is regulatory gene, and this regulatory gene produces or synthesizes its own product. And from this product decreases. This product decreases expression of structural gene. This product decreases the expression of structural gene, then at that time it is known as negative control. When the product of regulatory gene decreases the expression of structural gene, at that time we have negative control. And at that time the name of the control is repressor. The name of the control, the name of the product here is repressor. That means, dear students, regulatory gene produces the repressor, which decreases the activity, which decreases the expression of the structure. This is known as that is negative control. Are you getting me? Now, what happens in positive control? This is the regulatory gene, and this regulatory gene synthesizes its own product, and from this product there occurs increase expression of structural gene. When a regulatory gene synthesizes its product, and this product increases the expression of structural gene, at that time we call it as positive control, and at the positive control the product is known as inducer. That means this inducer increases the expression of structural gene in case of positive control. Now, dear students, on the basis of regulatory gene and on the basis of structural gene, this regulatory gene controls the activity of the structural gene. Clear? Now, when this regulatory gene represses, when this regulatory gene represses the activity of the structural gene, at that time we call it as negative control. And when the regulatory gene increases the expression of structural gene, at that time we call it as a positive control. Dear students, these are the various types of the genes are depending upon the control and depending upon the function. Now, dear students, this was about today's topic, that was gene expression. Now we will meet in next lecture. Till then, goodbye.